Okay, I think we are ready to go now. Welcome again to our webinar. My name is Victoria and I am your host for today. Before we start, I will give you a few remarks for your information. Please feel free to type in any questions that come up during the webinar in our questions box. You should see it at the top or right side of your Microsoft Teams window. The icon looks like a speech bubble with a question mark and it's named Q&A. We will answer as many questions as possible at the end of the webinar, and if there are too many questions, we will answer everything afterwards via email within the next 2 days, and we will get in touch with you. This webinar is also being recorded for you, so you will automatically receive the video within the next days by email as well. Now, I would like to introduce you to our today's speaker, Dr. Mohamed Bousbib. Mohamed holds a PhD in chemistry from Eötvös Loránd University in Budapest. He is a member of the Niche Analyzing and Testing family, contributing to the development and support of the software NEO team. Throughout his career, Mohamed has published numerous articles in prestigious peer-reviewed journals. So welcome, Mohamed. We are happy to have you here today, talking about using the Kinetics Neo software to determine the shelf life of potassium clavulanate in the pharmaceutical industry. I will now hand over to you to get started, so enjoy our webinar and have fun.
Thank you very much, Victoria, for your kind introduction, and a warm welcome from my side to everyone joining our webinar today. So today our webinar will focus into how to determine the shelf life of drugs and the final formulation by using Kinetics Neo software and the comparison between our method and the real-time and accelerated stability testing. Also, I would like to show you today how we can reduce the time from few years for real time and from few weeks accelerated stability to just few days by using our approach. So during this presentation, I will provide you a brief introduction to kinetic chemistry, including the definition of kinetics and the key factors influencing the reaction rate. I will also show you how our Kinetics Neo software can help you and how user-friendly it is and what type of data you need in order to perform the right kinetics analysis. And finally, I will show you, or I will review with you, the shelf life of potassium clavulanate by using our approach. So what is kinetics and how we can define the kinetics? So basically kinetics is based on two main aspects. The first one is to better understand the mechanism of a chemical reaction and to also describe the experimental data with a mathematical model that fit with the experimental data. And second part, we are using this mathematical model in order to predict the behavior of the material in a time comparative which is not easy or impossible to access. So actually, we have many factors which influence in the reaction rates in chemical process, or what we are calling them the external parameters: temperature, pressure, UV light or light generally, catalyst, solvent, and also the surface area of reactants. So as you can see that I have already highlighted in bold the temperature, pressure, and UV light because they are already implemented in our software. And for example, when we are speaking about the pharma industry, that it will be really helpful for them to study the kinetics of photodegradation or drug stability by using the light exposure. The catalysts and the solvents are generally included into the formal reactant and the product. Thermal analysis kinetics is based on the thermal analysis technique, which is like TGA, the thermogravimetric analysis, the differential scanning calorimetry TSC, and the dynamic mechanical analysis, which will help us in order to estimate the drug and the final formulation shelf life and the recommended storage condition or transportation. So when we are speaking about kinetics, the first things coming to our mind is to speak about the Arrhenius equation, but let's give you the general equation first. That the derivative of I will take my pen here. So we can take the derivative by the time of the conversion, which equal the Arrhenius equation multiplied by the reaction model. And the reaction model, it can be the first order, the second order, or autocatalytic reaction. So for Arrhenius equation or the Arrhenius dependence, so we have the A, which is the pre-exponential factor, which means in this case that the frequency factor, or which represent the frequency of collision between different reactants of the molecule. Also, we have the energy activation or activation energy, which is the minimum of energy what it need the reactant in order to transform from a reactant to the product. And also we have the other constant, for example, the R, which is the gas constant, and the T, the absolute temperature, which is the T in Kelvin, equal T degrees Celsius plus 273.15. So what is Kinetics Neo software and how this software can help us in order to determine our shelf life? Basically, the Kinetics Neo software is a tool for kinetics analysis and simulation of thermoanalytical data. And when we are speaking about the thermoanalytical data, that all techniques which are taken in consideration as a factor temperature. So in this case, we have the TSC, TTA, TGA, teratometer, ARC, and DMA. And we will focus about DSC, TGA, and DMA as they are the most used in the pharma industry. And that's why based on that data, we can import them in our software and we can do the kinetic analysis. So for kinetic analysis, there is two main approach or two main model, the model-based and the model-free. The model-based, it can be used for the multi-step reaction, and also, as you know, that in chemistry, that only 5% of the chemical reaction can be done by a single step, which is described by the model-free. So that's why we are using the model-based, which is crucial in this case in order to describe our chemical process. But sometimes a multi-step kinetic can be also described by a single step. This is when we have, for example, multi-step process, and we have it's dominated the rate of the single step with it, or it's limited by the rate of one step. So that's why sometimes even we have a multi-step model, it can be described by the model-free. After describing our model and checking the coefficient of determination, which is the R-square, which means the goodness of the fit, which is normally, if it's perfect, it's equal 1, or otherwise it can be closer to 1. So in this case, we can extract the different kinetics parameter, or what we are calling it kinetic triplet, the activation energy, the pre-exponential factor, and the reaction model. And based on that, based on this mathematical model, we can do the prediction at different heating rate, for example, dynamic, isothermal, and as we are speaking about prediction and isothermal, we can determine the shelf life, sure, taking in consideration the specification limit. So this is a screenshot from our software, which show us the different parts of our software. So first we have the data import, the different data which we can import from the thermoanalytical techniques, the TGA in this case, for example, we have the mass loss from 100% to almost 20%, which is almost 80% of mass loss, a different heating rate from 1 to 2 to 2, 5, and 10K per minute. After that, we have the kinetic analysis. We can do the kinetic analysis in this case and base it on two approach, the model-free or the model-based. And as I said, that it can be for the single step, and the model-based is for usually for the multi-step. And as you can see here, we have the different model, which we can do different model, create different model. After that, we can click on model summary in order to do the comparison and choose which one is the best. Here we have also the model design, as we know in chemistry that it's not always the reaction A goes to B and B goes to C, only consecutive way, but it can be competing or independent. So that's why our software can help us in this case to create the model design which can fit with the experimental data. Also, after creating this model design, we can also extract the different kinetics parameter for each reaction type. For example, here we have step B go to C to D, and we can determine the activation energy, the log of pre-exponential factor, the reaction order, and the contribution. Here we have the adjust part, or what we are calling the manual optimization, that you can click here to increase the contribution or to decrease the contribution or to adjust the experimental data based of this manual optimization Also, we have the automatic optimization that you can by single click here, and our software can optimize for you your model with the experimental data just in few seconds. We have also the statistical comparison, which I said before, that we have, for example, different model, and after that we can click here. It show for us a table which show the different statistical part, for example, the R-square or the coefficient of determination. Here we have the last part. So after importing the data and doing the kinetic analysis, we can do right now the simulation and prediction. So we have the different type of prediction at isothermal at different constant temperature, isothermal lifetime, dynamic at different heating rate, or even climatic, which I will speak later on. And as we are speaking about the shelf life, so we can do the prediction at different isothermal, for example, at 30 degrees C for 60% of humidity, and in this case we can choose the first one in order to do our prediction. So we are speaking about what kind of data we can use. Yes, we know right now we can take the data from the thermoanalytical technique, but what we need exactly, we need at least 3 measurements at different heating rate or at different isothermal measurement, and by this case we can import them and we can create our model. If you are asking how we can choose which one, is it 1 or 2 or 5? Yes, generally from our side we are advising that more heating rate, 4 or 5, can improve the accuracy and the confidence in the result, because more points allow us for better detection of the deviation. Also, in the pharmaceutical part that we are speaking of, we are recommending that to use the slow heating rate to ensure the controlled and uniform condition for accurate kinetic analysis. So in this slide, I'm going to show you that we are using the model-free or the single step model by using the two different of thermoanalytical technique, which is DSC, and also the TGA. Here the mass loss and here the heat flow. And we can see that here we have only one single peak, and here we have only one single mass loss, which can be described by our model, and after that we can extract the different kinetics parameter. So how we can experimentally determine the conversion for TGA data or for DSC. So basically we can, for the conversion, it's equal the partial mass loss divided by the total mass loss, and based on that we can determine our conversion, which means that the reaction progress. And usually it's between 0 and 1, or we can do it even by percentage, between 0 and 100%. With the same way or with the same approach, we can also calculate the conversion for the DSC by dividing the partial enthalpy on the total enthalpy in order to get our degree of conversion, which is always between 0 and 1. So how we can determine the shelf life of our drugs or our final formulation? First of all, I would like to give you a definition about what is a drug product and what is the difference between a drug product and a drug, and also between an excipient. The drug product is a final form which combine a drug and an excipient, and we know that the drug is the active compound in pharmaceutical formulation that provide the therapeutic effect. Where the excipients are inactive substance included in the pharmaceutical formulation. It's usually in order to enhance the stability or to improve the taste. And the drug product is the combination between a drug and excipient, and sometimes we are calling it the active pharmaceutical ingredient, which is API abbreviation. So before starting, I would like to show you why we choose exactly the potassium clavulinate as API in our case. So firstly, because it's crucial in the pharmaceutical industry, primarily due to its role in combating antibiotic, and also right now the global market of its is quite expanding due to the growing prevalence of antibiotic resistance infection and raising awareness about antimicrobial. Also, as you can see, that first of all, we can do by using our TZ309 Classic, we can determine the thermal stability of our drug. So we are taking 10 as a mass 10.5 milligram of potassium clavulinate in open aluminum crucible at and we are heating it up to 600 degrees C at 10 K per minute. Under inert atmosphere or under dynamic nitrogen atmosphere. So in this case, we can realize that we have different mass laws. The first one -1, 5%, -15%, almost -40, and -8. So almost 4 degradation step. And the first one, it's by combining with FTI, we can detect that it belongs to the water. And the first degradation, which belongs to our drug, it's that one at 185, which is under nitrogen atmosphere. So we can see that our drug is stable up to 186 under inert atmosphere. So after that, we can do something more than to determine the thermal stability. Yes, right now we have KineticsNeo software, and by adding just few different heating rate, we can determine the shelf life. But what is the shelf life in this case? The shelf life is the period of time from the date of manufacture that a drug product is expected to remain within its approved product specification while stored under defined condition. Yes, we know that there is no generic method to estimate and to set the shelf life, but in our case, we are aware that there is a real-time stability, the accelerated stability, and our approach, what we are calling it the stress testing. So in this case, also we need to take in consideration the different influenced parameters, like relative humidity, gas atmosphere under inert or under oxidative atmosphere, the time temperature condition, the light, and the other ones. So this is the same as the previous one to determine the thermal stability of our drug. After that, we need to add, as I speak before, that we need at least 3 heating rates. So in this case, we did 4 in order to increase the accuracy of our kinetic analysis and also to increase the accuracy of our prediction. So after doing the different 4 heating rates, we import them in our software. This is a screenshot from our software. So we choose here to do 1, 3, 5, and K per minute. And as you can see, also by using our software, we can do even the preparation. And here, the first peak which belongs to the water, it's already removed in order to take in consideration only the degradation of our drug. So after importing the data, so how we can determine the number of steps. So the easy way is just to visual inspection of the curve. So we can see that we have the first mass loss, the second mass loss, and the third mass loss. But in this case, is it true that we have only 3-step process? So that's why sometimes we need to do more investigation, and by using the DTG measurement, at 1 k per minute, and as you know that the TGG is the first derivative of thermogravimetry analysis, we can see that we have three main peaks, which is that one, and after that we have two shoulders. So we are also expecting that it can be 5 steps. So that's why after that we will choose the model with 5 steps with an order. And here, as you can see, that the fit or the model, it was quite perfect in this case, as the R-square it shows 0.999, which is almost 1. So after describing our experimental data and reaching the best, most suitable model, so in this case we can extract the different parameters, the activation energy, the N order, the contribution, and all of the stuff, and then we can go to the last step, which is the prediction. So for the prediction, I would like just to take example to show you that, for example, let's make a prediction for 5 years of degradation of drag under nitrogen atmosphere between 20 and 80 degrees C. So we can see here that, for example, if we make the specification limit here, that we can realize that for 70, for example, that we have a shelf life, it's 4.6 years. Yes, you will tell me that this specification limit is quite low. It's less than 0.01%. I will tell you, yes, that's correct. But in this example that we were based in nearly ideal condition without taking in consideration the other external factor, for example, the oxidative condition or under air or oxygen or the influence of excipients like amoxicillin or the effect of the relative humidity. Also, recently I was reading and I found many of articles and reviews they are publishing that they are using Arrhenius equation, and after that they are making a linear approach, which is not correct. As you can see that here we try to increase the temperature between 80 to 150, and we can see that we don't have a linear trend. It's no linear trend, which is show the accelerated degradation at higher temperature, and here it's clearly it show that it's going by high speed, and after that it decreases, which is almost exponential degradation. So step by step breakup. So we can basically, in order to determine the shelf life, that we need only a TGA instrument, and we can take few milligrams from final formulation or our drug, for example 10 milligram, and we can do the measurement at different heating rates. The minimum is 3, and under it's up to you, it's under atmosphere or under presence of air or oxygen, and then based on that we can import the data into our software, and we can create the most suitable model with the different reaction step in order or catalytic or any other reaction type, which already we in our software, or all of them is already included and implemented. And after that, after creating the most suitable model, that we can based on that to do the prediction of the shelf life. The average duration of our measured data for the TGA and kinetics analysis, it's already 5 days, so we are expecting that we can do 3 different heating rates, each one per day, and based on our kinetics software, you can do just in few minutes or few hours, you can create your model based on the complexity of the result, and also the duration of the measured data under TGA. It's also depend of is it slow heating or high heating. So here it's a comparison between the different testing methods. So we have the real time, which is it's the most accurate and reflect the actual storage condition, and it's required for regulatory approval. But the disadvantage is time consuming, it's taking months or even years, and too quite expensive. And we have also the accelerated stability, which is just taking few weeks, but the result or the extrapolated results may not always reflect the real condition. And what we are believing is the last approach, what we are calling is stress testing. So it's quite faster, more efficient, and provide you a deeper insight into stability, making it the most effective. And also based on this method that it will give you an insight or at early stage of the drug or the formulation during the development of the stability prediction. Also, we have another feature, what we are calling it the climatic prediction. As we all know that the climatic zone as defined by ICH, which is the International Council for Harmonization. So it's provide us ICH stability guideline, which provide us a general recommendation for stability testing under predefined climate zone. For example, the zone 1 is temperature, and we have between 21 and 45% of relative humidity. The Mediterranean one, the hot dry, the hot humid, and the last one, hot to higher humid. And in our software, the good things on it that we are not taking each region and we are in constant temperature, for example, at 25 and 60, and we are aware that it's not true. But our software, it's based on almost 30 years data, and it's globally at more than 100 locations worldwide, and using the real-time data, which can help you to do the precise prediction for your API stability. And this is some screenshot from our software or climatic prediction. And we can see that in the different regions, for example, Boston, United States, Australia, and Malaysia, and India. And we can see, for example, that during the months between June and August, or during the summer in United States, that we have higher temperature, but during the winter, or between June and August for Australia, it's quite low, or it's winter time right now there. And after that, we can see for Malaysia how it's quite the same the whole year, but we can see the difference here. For example, why there is this variation. This variation, it belongs to the day and to the night temperature. So here we can take it the day temperature, and here the night. That's why we have this kind of variation. And here in India, that we have this quite different data. So it's because it's influence of the monsoons. So basically, Kinetics Neo fulfilled the ICTAC Kinetics recommendation, and ICTAC is an international confederation for thermal analysis and calorimetry. How to get a trial version? So just go to kinetics.niche.com and click on the request trial version, fill the form, and in few days you will receive your software. And after that, we are always advising our customer to go to the learn section, and there is many example and training guidelines which can help you to explore our software and to know how our software is the capability of our software. And if you find any problem, or if you have any question or difficulties, you can reach us at kinetics.neo@niche.com. Also, I would like to take a moment to say thank you to Dr. Mukina Elena and Claire Strasser for their support and contribution. And thank you so much for your time and attention. And for further questions, please contact us on kinetics.neo@niche.com or at webinar_ngb@niche.com. And with that, I will pass the floor back to our moderator, Victoria.
Thank you so much, Mohammed, for this great presentation. We are still here for a few minutes to answer your questions. So if you have any, you can let us them know in the Q&A area. It is looking like a speech bubble with a question mark. You can type in all of your questions there. And Mohammed, I think we take a look at them together. I would directly start with the first one. Can we only use data from niche instruments, or are other instruments also possible? Can you say something about this?
Yes, sure. We can use the data from our niche instrument and also from no-niche instrument. Concerning the data, we need only 3 columns: time, temperature, and signal, and at any order for that. So basically, we can use for both for niche and no-niche instrument in order to do the prediction for the shelf life.
Great, thank you. Then the next question. By using your approach, how much time will it take to determine the shelf life?
Well, in order to determine the shelf life, that we need basically the data from the thermoanalytical techniques, and it will take, if we are speaking about at least 3 heating rate, it will take at least 3 days. And also by using our software to do the kinetic analysis, it will take around at least 4 hours. It depends on the complexity of the chemical process. So we can say that the average is 5 days, and we can reach or we can determine the shelf life for that.
Great, thank you. And the next question. Is Kinetics Neo software compatible with Windows 11? We are using this system in our lab.
Yes, our Kinetics Neo is compatible with Windows 11 and Windows 10 for 64-byte.
Okay, great. That would it be. I think there are no more open questions for now. So I would say, Mohammed, we wrap it up for today. Thank you for your great presentation, and to all attendees, you can see all of our email addresses on your screen. So if you also have further questions after you receive the recording, or if you use the trial version of Kinetics Neo, whatever, you can always reach out to us. So thank you for your attendance, and I hope that we see each other soon in another webinar. Enjoy the rest of your day. Bye-bye.
Thank you.